Okay, hello everyone. So I'm Michael Bradshaw. I'm actually a newly minted PhD as of Tuesday. And I've done a slight title change. So instead today we're going to be talking about the effects of biological knowledge graph topology on embedding-based link prediction methods. It's just the new and improved version of the original abstract. So this is Jane Doe, also known as MyGene2 patient 1930. Now, Jane seemed like a perfectly normal, healthy child until she began walking, which doctors described as gait ataxia, or abnormal but not due to muscle weakness. Shortly after that, Jane began having seizures, then developed a tremor and pancreatitis. Now, the weird thing about Jane's phenotypes or symptoms is that there's no obvious one disease that relates them all to each other. So Jane has what we know as a rare and undiagnosed disease. Now, paradoxically, rare diseases aren't actually that rare. It's estimated that somewhere between 25 and 30 million Americans are affected by a rare disease. On top of that, it's estimated that 72% of rare diseases are of genetic origin. So in a case like Jane's, what would typically happen next is a sample of her DNA would be taken, we'd walk through the sequencing and processing steps, and we'd use a variety of computational tools to identify variants. From there, the variants that need to be addressed or looked at more carefully in a case like this would be anything that is de novo or potentially rare recessive alleles that have been inherited from both parents. Now, this results in quite a large number of variants that need to be looked at, actually. There's anywhere from 44 to 82 de novo variants in each person's genome. However, we can quickly filter out anything that is known to be benign or falls into non-coding and otherwise ununderstood regions of the genome, but that still leaves us with a pretty sizable handful of variants that we need to investigate. So from here, what would typically happen is then you would look in the biological databases to see if there's any known connections between the genotypes and the phenotypes of the patient. However, what happens when there's no known connection between the two? At this point, these variants become what we know as variants of uncertain significance, or VUS. Now, just because we don't know of any direct connections between these genes and phenotypes doesn't mean we don't know anything about them. We can actually use the wealth of information encoded in biological databases, ontologies, and knowledge graphs to try and make inference about rare disease cases like Jane's. So we can overlay this information onto a knowledge graph, and suddenly it's quite apparent that there are potential indirect connections between the variants in someone like Jane and her phenotypes. Now, within the realm of learning on top of knowledge graphs, a recent advancement that's been adopted quite widely in the drug discovery space are knowledge graph embedding methods. Now, the way a knowledge graph embedding method works in a situation like this is that every node in this network is going to get encoded into a low-dimensional vector representation, where each node, let's zoom in on this red gene that we still have highlighted, is going to get represented as a series of numbers that describe where this node is located in multi-dimensional space relative to the other nodes in the network. From there, we can then perform a link prediction or guess which of these edges that don't exist should exist using some type of objective function, where we can take the other embeddings representing the other nodes in this network and then calculate, so look at those potential connections we're interested in, and then generate a score describing the likelihood of those edges actually existing. From there, it's simply then looking at whatever the highest number of edges are, and then plucking that out as the one that we're going to predict that there should be a link in between. Now, as I mentioned before, so this type of technology has been used quite successfully in the realm of drug discovery for predicting protein binding interactions or drug to disease or drug to gene interactions. However, we've really yet to see these methods be widely used and adopted into the methodologies for rare disease variant prioritization. Now, before that can really happen, I think that there's a couple technical limitations of these technologies that need to be addressed, namely node degree bias. Now, for a bit more context about how these networks are built, when it comes to the protein-protein interaction networks, the way these are built is they originate from protein binding experiments. Now, typically the way that these experiments run is we have one protein of interest, let's say TP53 in this case, And then we attempt to bind TP53 against dozens or hundreds of other proteins. And what this results in is a network that looks something like this, where TP53 gets dozens or hundreds of new edges, and everything else receives at most one. But then obviously this wasn't reflective of all of the proteins in the humans. So then we aggregate more experiments that were run the same way. So we incorporate then BRCA1 that was run in the same way, where BRCA1 got dozens or hundreds of edges and everything else got at most one. And as you continue to expand out, you can see how this problem just gets more and more exasperated. And this culminates in a degree distribution of the nodes of this network that looks something like this, where the overwhelming majority of nodes have relatively few connections, like the median of this distribution here is 100, but then there's the hub node here that has 5,872. So it's quite skewed. Now, this specific distribution is what we know as a power law distribution. So one of the characteristic features of that is when you plot it on a log-log scale like this, you can see this kind of like straight line forming right there. Now, a super awesome recent paper by Lucetta et al. Recently showed that as you aggregate more of these small protein binding experiments, that these power law distributions become virtually inevitable. As you aggregate 100 of them, 53% of the time it's going to form a power law distribution. When you aggregate 200, you've got 75% of the time getting that power law. And then when you're aggregating 300, we're seeing that 83% of the time the power law forms. So it really becomes virtually inevitable. Now, from this, we see how study bias, or our hyperfixation on just a small subset of genes and proteins, results in topological imbalance, exhibited through those power law degree distributions. Now, this shouldn't really be surprising, given that we know that 19% of genes receive 50% of attention. So that's 50% of experiments, grants, and publications are about this extremely small set of genes and proteins. Because of that, we also now know that the connectivity of these networks isn't necessarily representative of just biology. Rather, it's more reflective of our interest in genes and proteins as a community. But why does this really matter? Like, who cares if there's topological imbalance in these power law distributions going on? Well, it turns out that when we then train something like a knowledge graph embedding model on a topologically imbalanced dataset, it results in node degree bias, which is the tendency for a model to predict the links related to a gene proportional to its degree. So you can see that really well in this figure I've taken from a Bonner paper, where the degree of a node and its predicted score are extremely well correlated, such that even if a gene isn't actually connected to it, it's always going to rank well just because it has a high degree. So from this, we learned that high degree pretty much always equates to a high score. Now, this really becomes problematic when we think about how the predictions of our models are supposed to be used, where the whole point is we're using these to develop hypotheses to inform experimental results, which if we're then using that as intended, that's going to result in more experiments being generated about this already well-studied and well-characterized set of genes and diseases, which will then result in more topological imbalance and more node degree bias restarting over and over again. Now, this is what I call the bias cycle. And since we know that, okay, there's certain things that are always going to rank well in these models because of this cycle, it begs the question of what's being deprioritized by these same systems. And furthermore, what I want to know is how are these models performing on rare diseases, which is a class of diseases that have been historically understudied and are often lowly connected within these models. So to answer these questions, I needed a knowledge graph. So I'm using the Monarch knowledge graph here, which is arguably one of the larger, higher quality ones out there. And it's absolutely enormous. It has 500,000 nodes from 36 different sources of information. So other databases, ontologies. So this includes everything from disease, anatomy, phenotypes, including drug and chemical interactions, and not just for humans, but for a variety of model organisms as well. And it's very well connected with 11.5 million edges. And then for my methods so far, we're doing a pretty standard test train split of 80-10-10 and using a trans-E knowledge graph embedding method to start with. And then the question I'm going to be getting at is how well are these held out test set edges going to be ranking in a link prediction task, but specifically when we stratify them into 5 groups of genes and diseases. So we're looking at a group of heritable cancer genes, so this group of genes that's been historically well studied and well understood, at least relative to everything else in the human genome, in addition to a group of non-cancer related genes, and then 3 groups of diseases, rare diseases, ultra-rare diseases, and a group of control diseases. Now, the precise experiment we're going to be running is for each one of the nodes, be it a gene or disease in this network, we're going to look at each one in each group, and then we're going to look at all potential target nodes for it. So in this example, we'll say we're looking at a gene to a variety of diseases. Then we're going to score every possible one of those edges. So we're going to score every possible edge, and then we're going to rank them according to those scores, and then normalize our ranks between 0 and 1, where 0 would be the absolute worst and 1 would be the best. And then we're going to mask and pull out just those edges that we know actually exist because they were in our test set, and then look at the distribution of those normalized ranks. The key metric we're going to be looking at is the median normalized rank, which you can see is plotted here with the red dashed line. So you can see in this distribution of heritable cancer genes that it has a median normalized rank of 80. Now, as for the results, so what I found is that compared to the heritable cancer genes, that these other 4 classes of genes and diseases actually perform quite poorly in a link prediction task. So we're seeing kind of like that first example of the bias cycle coming out, where these things that have been historically well studied, well funded, and well understood rank much better in situations like this. Now, this model I'm using, TransE, is one of the original knowledge graph embedding models. And since it's been published, many more better and more complex models have come out. So we tried the same experiment with 2 more models, a rotate one and a complex one. And while the results improved some, we still seen this same general pattern where compared to the heritable cancer genes, these other 4 groups of genes and diseases really lag behind in terms of predictive performance. Now, returning to the structure of our knowledge graph. So we're only actually using 2 types of information for our tests. We're dealing with diseases represented as Mondo terms, or the monarch disease ontology, and then we're representing the genes and proteins with HGNC, or the Hugo gene nomenclature, which is only 2 of the 36 different types of information or meta nodes that are in this knowledge graph. Now, it seems that one of kind of like the unspoken assumptions of knowledge graph construction is that bigger is better. The more information we can aggregate, the better we're going to make inference about understudied entities like rare diseases. But as far as I know, this assumption has never really been tested, and it's certainly not been tested within the context of knowledge graph embedders. So that's what I want to know is, if we strip out all of the information other than what's absolutely necessary for the task at hand, how well does it do? So I created a drastically filtered down version of the knowledge graph that contains just Mondo and just HGNC, which results in like a graph that's about 14% of the size of the original one. And then re-optimized and retrained those models. And now we're going to look at what happens when we train our models on these filtered knowledge graphs. And the results are that in some cases, we can do substantially better using the filtered knowledge graph over the full one, which you can see here, where here on the x-axis, we're plotting the median normalized rank of models trained on the full graph, and on the y, we're looking at models trained on the filtered graph, where the diagonal line is a slope of 1 going through the origin of the graph. So if points are falling above the line, that means they do better with the filtered. If they're falling below the line, they did better with the full graph. Now, what we can see here is that for the gene groups, for non-cancer and cancer ones, they did universally better with the filtered knowledge graphs. And in 2 out of 3 cases, the ultra-rare diseases also performed better when we were using the filtered knowledge graph, with control diseases pulling up slightly better when we use the rotate plus filtered knowledge graph. So it seems from this, we've kind of contradicted that assumption that using all possible information is actually beneficial to predictive performance. An added benefit of this is the drastic reduction in the amount of time required to train and optimize these models. For example, with the full knowledge graph embedding models, some of these took as many as 30 days of runtime and on an NVIDIA A100 GPU to optimize and train, which if you were renting this model from something like Google Cloud, using just one of those chips for 30 days would cost you around $4,000, which is quite a bit. And over the course of this experiment, we ran 40 models, which you can see just how quickly like that could add up in cost. However, optimizing and training on the filtered knowledge graph, we were able to finish off all of those processes in under 1 day of compute time on the same hardware. So it looks like by filtering our knowledge graphs, we can both save time and improve performance and really enable more and better research. Now, returning to our knowledge graph structure, so going from 36 sources of information down to just 2 is a drastic reduction. What I want to know is just take that first step into exploring how these other pieces of information really contribute to predictive performance. So I started by looking at just the 22 other meta nodes in this graph that actually integrate and are related to Mondo and HGNC. So what I did is I reintroduced them one at a time. So I created 22 new filtered knowledge graphs that have 3 sources of information: Mondo, HGNC, and then 1 additional piece, and then re-optimized and retrained a model for each one of these. And the results I found is kind of a mixed bag, where in some cases, reintroducing these pieces of information made a good positive impact on performance, but not in all cases. So to highlight the winners of this reintroduction, it turns out when we reintroduced the human phenotype ontology, we saw the best performance for rare disease predictions, controlled diseases, and ultra rare, where we got a 0.24 increase in the median normalized rank for rare diseases, which is quite substantial, a 0.2 increase for controlled diseases, and 0.11 for ultra rare. We found some improvement for the cancer genes, where we got a 0.03 improvement when SGD, or the yeast genome database, was reintroduced. And then for non-cancer genes, we got an ever so slight improvement of 0.01 with Dictybase, or slime mold genome database, being reintroduced. Now, something that's important to note is that not all of these were actually beneficial. So you'll notice that there's the teal points on this graph, and the majority of time those are falling below the dotted line, or they're negative values, indicating that they were actually deleterious in terms of predictive performance. Notably for the ultra rare diseases, it turns out 17 out of 22 of these information sources actually decreased performance. Now, another thing that we learned from this experiment is that there seems to be no clear relationship between the quality or the quantity of information and the quality. If anything, it seems that the quality of information added by these seems to outweigh the quantity of information. So to illustrate that, if we look at the percentage of edges re-added to this graph relative to the filtered one, you can see here with HP, the human phenotype ontology, which had the best impact for the rare disease group. So that one added 130% more edges to the knowledge graph. Whereas NCBI gene, which still had a pretty good impact for rare diseases, but actually decreased performance for 3 of the 5 groups, added 150% more edges. So more doesn't necessarily make it better. And then a counterexample to that is then if we look at ChemB, which is a drug and chemical interaction database in ontology, you can see that this one increased the performance for rare diseases the second most by 0.23, so nearly the same as human phenotype ontology, but reintroduces only 90% more edges to the knowledge graph. So it seems to be that the quality or type of information encoded is more beneficial than the amount. I think given that the human phenotype ontology was expertly curated specifically to improve our understanding and communication around rare disease phenotypes, it really shouldn't be surprising that it benefited it the most in terms of predictive performance improvements. So in terms of best combinations, overall, the best 2 models that we've found are the filtered knowledge graph with HPO reintroduced, trained with the rotate model, and then rotate trained on the full monarch knowledge graph, where we actually found that the full knowledge graph is still slightly outperforming the filtered one with a median normalized rank of 90 and 93 for rare and ultra rare diseases. but closely followed by filtered that got 80 for rare diseases and 88 for ultra rare diseases. However, MNR doesn't really tell the full story. So if we zoom into just the top 10% of predicted edges, there's still potentially 2,000 genes or edges being predicted there. Now, if the end goal of these models is we're going to provide a list of potential new connections to clinicians or experimentalists, 2,000 is too many. Realistically, if you're not ranking in the top, let's say 100, that edge isn't actually going to matter or be relevant. So what I want to know is, like, if we zoom in and look at just the top 1 through 100, how many of the test set edges are we still finding? And when we zoom in and look at just the upper echelons of ranking, what we found is that the filtered model actually does perform better. It's identifying 1.6 times as many test edges at k equals 100 over the full knowledge graph model. However, we also note that for the ultra rare diseases, using the filtered one, they performed slightly worse than the full model. So there is a bit of nuance to this space still. Now, returning to the case of Jane Doe. So it turns out Jane has rare de novo mutations to 2 genes, NBEA and SSPO. So we searched for Jane's genotypes and phenotypes using our 2 best models and found something pretty encouraging, where in both cases, seizures in NBEA ranked extremely highly. It was ranked 11th and 13th by these models, which is about as good as you can hope for. That's pretty good. However, the next 3 highest ranked phenotypes in Jane's case all ranked quite a bit higher in the filtered model over the full model. So from this, big takeaways are that it seems that we've shown that knowledge graph embedding models can be biased against rare diseases. Additionally, we've contradicted the assumption that massively aggregating knowledge graphs is always beneficial. Bigger is not better. And I've also shown the importance of filtering your knowledge graph in terms of performance and also on runtime. And with that, I'd like to thank my collaborators, my advisor Ryan Layer and Alyssa Gaskill from the Children's Hospital. And you can find this work on bioRxiv right now. And with that, I'll take questions.
So thank you very much for this talk. Very, very interesting and very important. I have one question though. So when you have gene disease associations, that's what you predict. So you have a gene and you have a disease. And if that is all you keep, I have no idea what biological assumption you make in order to make any prediction whatsoever. Because if you have a rare disease, there is exactly one gene associated with one disease. And if it's ultra rare, then this disease is not associated with any other gene. It's Mendelian, right? So it's basically one gene, one disease. So unless you have something like shared phenotypes or something similar, like this is why HPO might help. Unless you have this, the only thing you can predict by is how often you have seen a disease in your training data. And then you just rank your diseases based on the frequency. You don't even need node degree. And you can predict, and you get a very good predictive performance if you simply rank independent of any other information. You don't even need genes. You just rank diseases based on the number of genes they're associated with, or take the number of publications they're mentioned in. And you get a predictive performance that is sometimes as good as some even very recent published results. So I think, in my view, it's not so much about the size. Maybe more is even better, but the important point is that you need a biological hypothesis that you're testing. And if you just put these two together in a knowledge graph whatsoever, you get a predictive performance, but there's no biological information in there to even make such a prediction. And this is why what you're measuring are different biases. I'm not sure this was a question, but but I think it's just a comment on what you're doing. And then there are ways to overcome this degree bias, because there are specific ways to incorporate degree bias, for example, in trans-E, because you have this degree bias because you see a certain entity more than once. So if you have 2 genes associated with a disease, you see this disease twice, in contrast to other diseases. So you could, for example, just sample all other diseases, if you have a disease only once, a gene disease association, just repeat it. So there are different ways, or there are more sophisticated ways to account for this. And I'm not sure, did you try any of those? Or there are also different ways in which you can split your gene disease association. You don't split them by the pairs and just randomly sample an edge, but rather you split by the gene or just split by the diseases. And then you sample your gene disease associations from those. So you withhold some information of one node type. So did you try some of these techniques?
We didn't, but you're absolutely right that those methods do exist. And there's like quite a good number of frameworks out there for the way in which you run your training, and being able to control for those biases are essentially what becomes just like a massive class imbalance issue. And it does seem that like if you're going to move towards using these types of models for something like rare disease variant effect prediction or variant prioritization, that you're really going to be needing to like address it from multiple different angles, from like the models you're using, the input data you're using, and additionally to your like training methodologies as you spoke to.
So please can move forward, because online participants, they need to also hear you.
I'm also not great at repeating questions, so I'd rather.
Anyway, I'm curious about your thoughts about the performance difference in full knowledge graph and sub-knowledge graph. You kind of implied that some ontologies make a positive contribution, some make a negative contribution. You kind of implied some of these ontologies have better quality, others are not as good. Do you have any other thoughts which might contribute to the cause of this performance difference, like how knowledge graph itself is constructed?
As for how the graph is constructed, no. Future work that I'd hope to work on is addressing like how we can quantify the quality of information in any source of data, which does seem quite messy. But I guess to your point of like re-adding information and it having like different effects, one thing we noted is that like depending on the class of disease or genes that you're trying to work on, that different data sources impacted it wildly differently. Like HPO improved performance for the disease groups, but really didn't have any impact at all when we looked at the gene, the gene groups. So it seems like depending, like it's context specific. Like we should be building our knowledge graphs for learning applications with a specific context in mind.
Thanks.
Yeah.
Hello. This isn't really my field, but I just had a question about whether or not you used drug averse effects or like off-target drug effects into these knowledge graphs to try to see whether when the gene is genetically altered, does that recapitulate any effects that were brought on by inhibitors or agonists?
Yeah, so I don't look directly at that question. However, there are several like of the ontologies or databases that are being incorporated that do address and encode exactly those issues.
Thank you.
Hi, I'm not from the field. I hope it's not a too naive question. I thought that some degree of study bias should be natural, right? So for example, breast cancer, maybe it makes sense that we research the BRCA genes a lot. And therefore also there should be some natural degree of topological imbalance. So my question would be if you can account, or how do you account for that your notion of under and over representation is not also biased in that way?
Yeah, so we don't address that entirely. However, to your point that like, yes, like we don't want to bag on cancer because it's been studied so much. It's really like we need to lift everything. It's not that we need to drag down the amount of information we have about cancer. It'd just be lovely if we had more information about everything else too.
Thank you.
One more. So I could build knowledge graphs as well. So I guess my takeaway is that bigger can be better, right? Because it allowed you to do that experiment of essentially picking subgraphs and testing them, right?
Yeah.
So I'm actually curious whether you have any thoughts on some more principled ways to select those subgraphs, right? You might not always have a great evaluation setting, right? Or maybe the task is complicated. So maybe we just need some methods for that, right?
Yeah, I think that's like a very clear future work for this, is that like we need methods for filtering or aggregating knowledge graphs and quantifying like what information will be useful. Because like you could easily take something like kind of like a feature selection approach where you just like add them back in one at a time and see how that improves things. But that's with how long it takes to optimize and train these models and how much information we have, that's not really realistic. One idea I've had about that is like, if we look at like the number of edges added to things of extremely low degree. So like, let's say like, if we add like HPO back in, how many diseases that previously only had a single edge do we bump up to having like 5 edges or 10 edges and quantifying it in that way? But I think that's like a very good open question.
And I guess for performance, for the graph embeddings, I recommend Rust. There's some packages out there like that to cut down some time. All right, thanks for your answer.
Yeah.
I have a question. We have, then after that, maybe one more question after that. There is a huge NIH project called Eliminating the Druggable Genome. It was a couple of years ago. So I'm wondering, because that project also aimed at finding not so highly studied genes and disease connections. So I'm wondering why you chose the knowledge graphs and the ontologies that you chose, and why based on what criteria did you choose the ontologies and knowledge graphs that you were looking at?
Yeah, that's a good question. So I'm using the Monarch knowledge graph and anything that was incorporated in it by their consortium. Is that what they technically are? But so I've been working in the rare disease space for the entirety of my time in graduate school, and they're like kind of like the leaders in building methods for that based on ontology approaches. So that's how I ended up with the Monarch knowledge graph. And then as for going down to using HGNC and Mondo, it seemed it was the available annotations for like the gene and disease groups I had were most readily mappable into those 2 data sources, as opposed to any of the other ones.
Yeah, I think it was human disease ontology that they used.
Yeah. Mm-hmm.
Any other last one?
I'm wondering if you have tried other splits other than 80-10-10.
So originally, I actually didn't want to use 80-10-10. What I believe works better is if you can version by time and look at like how these sources have grown naturally over time, I think is a better way to split. However, finding versioned copies of like something as large as the Monarch knowledge graph or really any of the ontologies can be a little bit difficult, at least like getting them to all match up over multiple years. So that's why I'm using 80-10-10, but I haven't looked at other ones.